The Genuine Article. Discussion and commentary based on articles from JACC.
Hello, I'm Tony DeMaria, the editor of JACC, here with Inside JACC. And at the moment I'm speaking with Bob Roberts, who's probably known very well to everyone. Bob is currently the CEO of the Ottawa Heart Institute, and he's the director of the Canadian Cardiovascular Genetics Center. And Bob is the senior author on a paper appearing in JACC of considerable interest that deals with the 9p21 risk locus for coronary artery disease. So Bob, why don't you just tell us a bit about the study that's in press?
I think the essence of the study indicates that the 9p21 genetic variant, which obviously everyone can have either one or two copies or none of, if you have one copy, your chances of having single vessel disease are much higher, as opposed to if you've got two copies, you have a twofold increased risk of having triple vessel disease as defined by coronary artery angiograms.
Well, you know, I should begin actually by congratulating you for describing the 9p21 variant was really one of the most striking genetic genotypes that deals with coronary disease. And I guess the question you were looking at in large measure was whether or not this variant predisposed to atherosclerosis or predisposed to myocardial infarction as a complication of atherosclerosis.
Yes, that's correct, Tony, because while everyone around the world now has confirmed that 9p21 is a risk for coronary artery disease and or myocardial infarction, we still do not know much about how it does it. So in this study, what we were looking for is, does it give really its mechanism to atherosclerosis, or is it, say, for example, thrombosis or plaque rupture? Our findings would strongly indicate that the predominant mechanism is through increased formation of atherosclerosis. When we took care of the number of vessels and then looked at myocardial infarction superimposed, it did not add to the increased risk. In other words, all of the risk appeared to be absorbed in the laying down of atheroma, and that this would say to us, since MI was no longer increasing that risk, that the mechanism is more likely to be atheroma rather than thrombosis or plaque rupture.
I was interested to read that you saw this relationship between the severity of coronary disease and the dosage of 9p21 in both younger people and older people, and in fact it related to left main disease as well.
Yes, I think it's important to point out that the strength of this paper in terms of supporting that 9p21 as a risk for severity or the number of vessels involved had 3 points to make. First of all, we did it in a young population, and the people were all blinded to the genotype. Then we replicated that in a second population that was older, both populations defined by coronary angiography. And thirdly, we then looked at two independent descriptors, the Duke and the Gensini method. All of them showed the same correlation, and all of them showed that if you had two copies of the gene, you are more likely to develop left main disease as well as triple vessel.
Well, Bob, I guess the last question, given all the data that's appeared regarding 9p21, do you feel now that it should be part of the routine evaluation for the coronary risk factor profile?
I think that's a difficult question to answer, because most people would say, if you can't do more about it, should you in fact really screen for it? And certainly we do not have anything at the moment that can prevent 9p21 because we do not truly understand how it does what it's doing. On the other hand, what we have done here is that people who have a family history, who want to be tested for it, we do so, we let them have the information, but we explain to them, all we can say to you is if you've got 1 or 2 copies of 9p21 at the moment is to increase the intensity whereby you're going to treat other known risk factors and lifestyle, because we certainly don't have anything specific to give regarding 9p21.
Well, thanks a million for this information. It's, I think, an important insight that will help future research to try to target specific preventive therapies for those who have the 9p21 allele. For Inside JACC, I'm Tony DeMaria.
The Genuine Article. Discussion and commentary based on articles from JACC.